Hi everyone, welcome to the video lecture for Unit 2, Module 3.2.1. In this one, we're going to discuss white blood cells. As always, here are the learning objectives, and this one is a little interesting because we need to talk about this in blood, but we also need to talk about this in immunity too. So we're going to go over a little bit of it here, but we're also going to talk about it when we get to our immunity section in the next unit. So some of these might overlap a bit, just a heads up. All right, let's get started. So unlike red blood cells, white blood cells, or WBCs as it's usually called in common text, have nuclei and other organelles, and they lack hemoglobin. White blood cells, or leukocytes, help defend the body against invasion by pathogens. They also remove toxins, waste, and abnormal or damaged cells. Several types of white blood cells can be distinguished microscopically in a blood smear by using either two standard stains, a Wright stain or a Kiesma stain. Traditionally, white blood cells have been divided into two groups based off of their appearance after staining. We have Group 1, which is granular leukocytes or granulocytes, with abundant stained granules, the neutrophils, the eosinophils, and the basophils. And then your agranular leukocytes, with few, if any, stained granules. They don't have that many, and which are the monocytes and your lymphocytes. This categorization is convenient but somewhat misleading. So in reality, the granules in granular leukocytes are secretory vesicles and lysosomes, and the agranular leukocytes also contain vesicles and lysosomes, but they're just smaller and more difficult to see with the light microscope. A typical microliter of blood contains about 5,000 to 10,000 white blood cells, compared with about 4.2 to about 6.3 million red blood cells. Most of the white blood cells in the body at any moment are in connective tissue proper or in organs of the lymphatic system, and circulating white blood cells are only a small fraction of the total white blood cell population. So, unlike red blood cells, white blood cells circulate for only a short time in their lifespan. White blood cells migrate through the loose and dense connective tissues of the body, and they use the bloodstream to travel from one organ to another for rapid transportation to areas of infection or injury. As they travel along the miles of the capillaries, white blood cells can detect the chemical signs of damage to surrounding tissues. When these cells detect problems, they leave the bloodstream and enter the damaged area. Circulating white blood cells have four characteristics. One, all can migrate out of the bloodstream. When white blood cells in the bloodstream are activated, they contact and adhere to the vessel walls in a process called margination. After further interaction with the endothelial cells, the activated white blood cells squeeze between the adjacent endothelial cells and then enter the surrounding tissue. This process is called emigration or diapedesis. So two, all are capable of amoeboid movement. Amoeboid movement is a gliding motion that's made possible by the flow of cytoplasm into slender cellular processes extended in the direction of the movement. So the movement is also named like that because it's similar to that of an amoeba or a protozoan. The mechanism is not fully understood, but it involves a continuous rearrangement of bonds between the actin filaments and the cytoskeleton. It also requires calcium ions and ATP. This mobility allows white blood cells to move through the endothelial lining and into the peripheral tissues. Three, all are attracted to specific chemical stimuli. This characteristic, called positive chemotaxis, guides the white blood cells into evading pathogens, damaged tissues, and other white blood cells. Then neutrophils, or group 4, neutrophils, eosinophils, and monocytes are capable of phagocytosis. These cells may engulf pathogens, cell debris, or other materials. Neutrophils and eosinophils are sometimes called macrophages, and to distinguish them from the larger macrophages in connective tissues, macrophages are monocytes that have been moved out of the bloodstream and have become actively phagocytotic. So, neutrophils, eosinophils, basophils, and monocytes are part of the body's non-specific defenses. A variety of stimuli activate such defenses, which do not discriminate between one type of threat and another. Lymphocytes, in contrast, are responsible for specific defenses, which mount a counterattack against specific types of invading pathogens or foreign proteins. So, about 50 to 70% of the circulating white blood cells are neutrophils. This name reflects the fact that the granules of these white blood cells are chemically neutral and thus are difficult to stain, either with acidic or basic and alkaline dyes. A mature neutrophil has a very dense segmented nucleus with 2 to 5 lobes resembling beads on a string. This structure has given neutrophils another name, polymorphonuclear leukocytes, or PMNs. Polymorphs, or polys as they're often called, are roughly about 12 micrometers in diameter, and their cytoplasm is packed with pale granules that contain lysosomal enzymes and bactericidal compounds. Neutrophils are highly mobile. They're generally the first of the white blood cells to arrive at the site of an injury. These very active cells specialize in attacking and digesting bacteria that have been marked with certain antibodies or complement proteins, which are plasma proteins involved in tissue defenses. Upon encountering a bacterium, a neutrophil quickly engulfs it, and the metabolic rate of the neutrophil increases dramatically. Along with this respiratory burst, the neutrophil produces highly reactive destructive chemical agents, including hydrogen peroxide and superoxide anions, which can kill bacteria. Meanwhile, the vesicle containing the engulfed pathogen fuses with lysosomes that contain digestive enzymes and small peptides called defensins. This process, called degranulation, is because it reduces the number of granules in the cytoplasm. Defensins kill a variety of pathogens, including bacteria, fungi, and enveloped viruses, by combining to form large channels in their plasma membranes. Digestive enzymes then break down the bacterial remains. While actively attacking bacteria, a neutrophil releases prostaglandins and leukotrienes. The prostaglandins increase capillary permeability in the affected region, contributing to local inflammation and restricting the spread of injury and infection. Leukotrienes are hormones that attract other phagocytes and help coordinate the immune system response. Most neutrophils have a short lifespan. They survive in the bloodstream for only about 10 hours or so. So, when actively engulfing debris or pathogens, they may last maybe 30 minutes or less. A neutrophil dies after engulfing 1 to 2 dozen bacteria, but its breakdown releases chemicals that attract other neutrophils to the site. A mixture of dead neutrophils, cellular debris, and other waste products form in the pus that's associated with infected wounds. Eosinophils have granules that stain darkly with iosin or red dye. The granules also stain with other acid dyes, so the name acidophils applies as well. Eosinophils generally represent about 2 to 4% of the circulating white blood cells. They are similar in size to the neutrophils. However, the combination of deep red granules and typically double-lobed nuclei make eosinophils easily identifiable. Eosinophils attack objects that are coated with antibodies, and they'll engulf antibody-marked bacteria, protozoa, other cellular debris, but primarily its mode of attack is the exocytosis of toxic compounds, including nitric oxide and cytotoxic enzymes. This is particularly effective against multicellular parasites that are too big to engulf, such as flukes or parasitic roundworms, things like that. The number of circulating eosinophils increases dramatically during a parasitic infection. Eosinophils increase in number during allergic reactions as well because they're sensitive to circulating allergens, materials that trigger allergies, obviously. Eosinophils are also attracted to sites of injury, where they release enzymes that produce inflammation that's also produced by the mast cells and the neutrophils. This controls the spread of inflammation to adjacent tissues. The basophils have numerous granules that stain darkly with basic dyes. In a standard blood smear, the inclusions are deep purple or blue. They measure about 8 to 10 micrometers in diameter, and basophils are smaller than neutrophils and eosinophils. They're also relatively rare, accounting for less than 1% or so of the circulating white blood cell population. Basophils migrate to the injury sites and cross the capillary endothelium to accumulate in damaged tissues. There, they are discharged like granules into the interstitial fluid. The granules contain histamine, which dilates blood vessels, and heparin as well, which is a compound that prevents blood from clotting. You may have heard that if you've been in the hospital. Stimulated basophils release these chemicals into the interstitial fluid to enhance the local inflammation incited by mast cells. Although mast cells release the same compounds in damaged connective tissues, mast cells and basophils are distinct populations with separate origins. Stimulated basophils also release other chemicals that attract eosinophils and other basophils to the area. Monocytes are spherical cells that may exceed 15 micrometers in diameter, nearly twice the diameter of a typical red blood cell. When flattened into a blood smear, they look even larger, so monocytes are fairly easy to identify. So you should be able to do that on the stains when we look at those in the lab. The nucleus is large and tends to be oval or kidney bean shaped rather than lobed. Monocytes normally account for about 2 to 8% of circulating white blood cells. And an individual monocyte is transported in the bloodstream it remains in circulation for about 24 hours before entering the peripheral tissues, and there it becomes a tissue macrophage. Macrophages are aggressive phagocytes. They often attempt to engulf items as large or even larger than themselves. And when active, they release chemicals that attract and stimulate neutrophils, monocytes, and other phagocytotic cells. I can tell you one of the earliest memories I have of seeing these things is if you've ever seen the Magic School Bus and they're in the episode where Ralphie is sick, the white blood cells are going to attack the bus and they're trying to engulf it. That is monocyte activity. Active macrophages also secrete substances that draw fibroblasts into the region, and the fibroblasts then begin producing scar tissue to wall off that injured area. And that'll take us into hemostasis, which we'll talk about a little later. So typical lymphocytes are slightly larger than red blood cells, and they lack abundant, deeply staining granules. In blood smears, lymphocytes typically have large, round nuclei surrounded by a thin halo of cytoplasm. They account for about 20 to 40% of the circulating white blood cells, and lymphocytes continuously migrate from the bloodstream through the peripheral tissues and back to the bloodstream. Circulating lymphocytes represent only a small fraction of all lymphocytes. At any moment, most of your body's lymphocytes are in other connective tissues and organs in the lymphatic system. So circulating blood contains about 3 functional classes of lymphocytes. They cannot be distinguished with a light microscope, so not something you need to worry about in the lab. Just know the functional difference here. Number 1, T cells are responsible for cell-mediated immunity, which is a specific defense mechanism against invading foreign cells, and for the coordination of the immune response. T cells either enter peripheral tissues and attack foreign cells directly or control the activity of other lymphocytes. B cells are responsible for humoral immunity, which is a specific defense mechanism that involves the production of antibodies. These antibodies are distributed by blood, lymph, and interstitial fluid and are capable of attacking foreign antigens through the body. Activated B cells differentiate into plasma cells, which are specialized to synthesize and secrete antibodies. The T cells responsible for cellular immunity must migrate to their targets, but the antibodies produced by plasma cells in one location can destroy antigens almost anywhere in the body. And then 3, you have the natural killer, or the NK cells, that carry out the immune surveillance and the detection and subsequent destruction of abnormal cells. NK cells, sometimes known as large granular lymphocytes, are important in preventing cancer. A variety of conditions that include infection, inflammation, and allergic reactions cause characteristic changes in the circulating populations of white blood cells. By examining a stained blood smear, we can obtain a differential count of the white blood cell population. The values reported indicate the number of each type of cell in a sample of 100 white blood cells. So the term leukopenia indicates an inadequate number of white blood cells. Leukocytosis refers to an excessive number of white blood cells. So a modest leukocytosis is normal during an infection. Extreme leukocytosis generally indicates the presence of some form of leukemia, which is a cancer of the white blood cells. The endings penia and osis can also indicate low or high numbers of specific types of white blood cells. For example, lymphopenia means too few lymphocytes, and lymphocytosis means too many. So stem cells that produce white blood cells originate in the red bone marrow with the divisions of the hemocytoblasts. As we've talked about before, hemocytoblast divisions produce myeloid stem cells and lymphoid stem cells. Myeloid stem cells divide to create progenitor cells, which give rise to all of the formed elements except lymphocytes. One type of progenitor cell produces daughter cells that mature into red blood cells. A second type produces cells that manufacture platelets. A third type produces daughter cells to develop into neutrophils, eosinophils, basophils, and monocytes. Granulocytes, again, which are basophils, eosinophils, and neutrophils, complete their development in the red bone marrow. These white blood cells go through a series of maturational stages, proceeding from blast cells to myelocytes to band cells before becoming mature white blood cells. For example, a cell differentiating into a neutrophil goes from a myoblast to a neutrophilic myocyte and then becomes a neutrophilic band cell. Some band cells enter the bloodstream before completing their maturation. Normally, about 3 to 5% of all circulating white blood cells are band cells. Monocytes begin to differentiate in the red bone marrow, and then into the bloodstream, and complete development when the process or when they become free macrophages in peripheral tissues. The process of lymphocyte production is called lymphopoiesis. Some lymphocytes are derived from lymphoid stem cells that remain in red blood bone marrow. These lymphocytes differentiate either into B cells or NK cells. Most of the lymphoid cells that produce lymphocytes migrate from the red bone marrow to peripheral lymphatic tissues, including the thymus, the spleen, and the lymph nodes. As a result, lymphocytes are produced in these organs as well as in the red bone marrow. Lymphoid stem cells migrating to the thymus mature into T cells. Factors that regulate lymphocyte maturation are incompletely understood. Until adulthood, hormones produced by the thymus promote the differentiation and maintenance of T cell populations, and the importance of the thymus in adults, especially with respect to aging, remains controversial at best. In adults, the production of B and T lymphocytes is regulated primarily by exposure to antigens or foreign proteins, cells, or toxins. When antigens appear, lymphocyte production escalates. Several hormones called colony stimulating factors, or CSFs, are involved in regulating other white blood cell populations. Four CSFs have been identified. Each stimulates the formation of white blood cells, or both white blood cells and red blood cells. You have the M-CSF, which stimulates the production of monocytes. G-CSF stimulates the production of granulocytes, again, neutrophils, eosinophils, and basophils. You have GM-CSFs, which stimulate the production of both granulocytes and monocytes. And then you have multi-CSF, which accelerates the production of granulocytes, monocytes, platelets, and red blood cells. Chemical communication between the lymphocytes and other white blood cells helps to coordinate the immune response. For example, active macrophages release chemicals that make up lymphocytes or that make lymphocytes more sensitive to antigens and also accelerate the development of specific immunity. In turn, active lymphocytes release CSF and GM-CSF, reinforcing nonspecific defenses. Immune system hormones are typically under intensive study because of their potential clinical importance. The molecular structures of many of the stimulating factors have been identified, and several can be produced by genetic engineering. The USDA approved the administration of synthesized forms of EPO, GCSF, and GM-CSF to stimulate the production of specific blood cell lines. For instance, a genetically engineered form of GCSF, sold under the name of filgrastim or Neupogen, is used to stimulate the production of neutrophils in patients undergoing cancer and chemotherapy. The rest of these slides here are just to give you a brief overview of everything we talked about and just for your personal review.